You might be wondering about now, how do scientists measure reaction rate? Well, to measure the reaction rate, you need to be able to measure the concentration of at least one component in the reaction mixture at any point in time. Some reactions occur slowly enough that samples can be periodically withdrawn from the reaction vessel and analyzed to determine the progress of the reaction. But this is a tricky situation because a lot of reactions are very fast and the reaction may continue even after the sample has been withdrawn. So you have to stop the reaction's progress when you withdraw that sample, ideally. There are three techniques that are commonly used to monitor the reaction mixture: polarimetry, spectroscopy or spectrometry, and pressure measurement. The following three techniques are often able to be utilized as a continuous monitoring technique where the reaction can actually be done inside the spectrometer or spectrophotometer or other reaction vessel that allows the measurements to be taken while the reaction is being performed. The first of these techniques is polarimetry, which measures the change in the degree of rotation of plane polarized light caused by one of the components over time. This one is limited in use to substances that actually rotate plane polarized light, which means they have to be chiral. We haven't discussed chirality in this course yet, and maybe a little bit last semester, but we'll try to get to that by the end of the semester. The next technique is spectrophotometry, which measures the amount of light at a particular wavelength absorbed by one of the components, or it could even measure a light that's given off by one of the components. And the next and last is total pressure. The total pressure of a gas mixture is stoichiometrically related to the partial pressures of the gases in the reaction, as we saw in Chapter 6. Sometimes it isn't possible to measure the concentration of one of the components without withdrawing a sample of that component. So aliquots can be taken at specific time intervals during the reaction and then an analysis performed. For example, a titration can be done or a gravimetric analysis, which means to use a scale to weigh the sample. Also, gas chromatography can be used to measure the concentrations of various components in the mixture, assuming that you have an internal standard for that. And these are for samples that have volatile components because gas chromatography is limited to components that are relatively volatile. And you can separate the mixture by the sample's affinity for the surface of the column. Here's a simple example of a spectrophotometer that can measure the absorption or emission of light at a particular wavelength by the sample, which can then be correlated to concentration. Here we have a source of light which goes through a slit and then a monochromator. The monochromator is what determines the wavelength of the light that will be measured. And then that particular wavelength passes through the sample and is absorbed, and the amount of light that is absorbed is detected by the detector and then ultimately recorded by the computer. Here's a simplified example of a gas chromatography instrument where an aliquot is injected into a heated injection port, and that is why the sample needs to be volatile for this method to be useful. A carrier gas, which is usually helium, then carries the sample through a column, and the column separates the sample into its individual components where it's detected by the detector, and that detector then transfers the information to a computer in the form of a chromatogram.